Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on generation and conduction of nerve impulse. Now, in this lecture, we are going to discuss about how the nerve impulse is generated. That is the first part of our discussion, and in second part, we are going to discuss how the generated nerve impulse is conducted inside the neuron. Okay, so how the nerve impulse propagate throughout the plasma membrane of the axon. Now here, this one is the neuron, and this is the stimulus. And with the red color, as you can see, depolarization is shown. That means at this point they are showing that the nerve impulse or the action potential is generated. Now we can imagine this situation like this. Since this is the stimulus, so imagine that you are standing at some place with your friend, and your friend want to show you something, but you are not looking at that point where your friend want to show you something. Now what your friend is going to do that with his hand, with his finger, he is going to touch your hand. Okay, so when your friend your touches your hand, that time you realize that your friend has touched your hand. And why you are realizing that? Because in your hand now the nerve impulse has generated, and the nerve impulse has propagated through the sensory nerve in the spinal cord. Okay, now nerve impulse from your spinal cord, it will reach to the brain center via the ascending track. So from your friend is touching your hand, so the point where your friend is touching your hand, from that point the nerve impulse is generating, and nerve impulse propagate through sensory nerve to your spinal cord, and then through spinal cord the information or the input or the action potential reaching to the brain center. And that time you are realizing that your friend want to tell you something. Okay, so this is how the this is the basic that we need to understand. The stimulus is responsible for the generation of a nerve impulse. Okay, and here in this figure you can see how the nerve impulse is propagating. Okay, so now what are this stimulus? Okay, what is the definition of stimulus? So stimulus can be defined as a sudden change in the environment. Okay, any change in the environment suddenly, that can be a stimulus. And when we say environment, that means the external environment or inside our body. Okay, so any sudden change can excite. If the sudden change or the stimulus is strong enough, so the stimulus can excite our nerve fiber, our neurons, or our nerves. Okay, or if the stimulus is strong enough, so it can it can excite our muscle, or the stimulus as a whole, the stimulus can stimulate our body. Okay, so in short, the stimulus is responsible for the excitement of our nerve cell or neuron, and now the nerve impulse is generated if the stimulus is strong. So this strong stimulus are known as threshold stimulus or adequate stimulus. And if the stimulus is not strong and the stimulus is not exciting our nerves, or it is not generating the nerve impulse in our nerve, that time we can say the stimulus is sub threshold, or the stimulus is the inadequate stimulus. Now we can classify stimulus into four category. One is mechanical stimulus, second is physical stimulus, and for example the heat or the cold or the humidity, all this factor can be responsible for the generation of nerve impulse. Then some chemical stimulus, some chemicals are responsible for generation of nerve impulse. For example, as some acid or base, they are strong enough to generate the nerve impulse. Okay, and finally the electric current can generate the nerve impulse. So these are all the stimuli which generate the nerve impulse. Okay, now we are moving forward. Now we are discussing the third point. Okay, now here, now we are discussing resting membrane potential. Now, what is resting membrane potential? What is the meaning of this resting membrane potential? Okay, so see, uh, resting membrane potential is the potential when the nerve impulse is not propagating through the neurons. Okay, that is called resting membrane potential. And how we can measure this resting membrane potential? So we can measure the resting membrane potential with the help of voltmeter, and as you can see there are two electrode. One is recording micro electrode and second one is the reference electrode. So these two electrodes, they are connected with the voltmeter to measure the resting membrane potential of neuron, okay, in which the nerve impulse is not propagating. Okay, so now what is done for measuring this membrane potential? See, the tip of the recording micro electrode is inserted inside the neuron. Okay, and the reference electrode is placed in the extracellular fluid. So in the figure, as you can see, uh, this is the plasma membrane of neuron. This is the cytoplasm they are showing. Okay, and as you can see, the recording micro electrode, the tip, so the tip is immersed in the cytosol. Okay, and this reference electrode is placed on the outer surface in the extracellular fluid. So as you can see, when we are measuring the resting membrane potential, that time the voltmeter, the indicator of voltmeter is showing minus 70 mV. So this minus 70 mV is known as, is called as resting membrane potential. Minus 70 mV is considered as resting membrane potential. Okay, and the minus sign indicate that inside the neuron more negative charge is present comparing to outside. So here, as you can see, on the inner surface of plasma membrane there is a presence of negative charge inside. Okay, so this is about the resting membrane potential and how the resting membrane potential is measured. Okay, and here on left hand side in this figure, see, this is the plasma membrane of neuron. This is the positive charge is present on outer surface, inner surface negative charge is present. Now in extracellular fluid, see, outside the neuron extracellular fluid is present, and in this fluid both positive and negative charge ions are present, and as well as in cytosol also positive and negative charge ions are present. Okay, positive ion and negative ions are present in cytosol and extracellular fluid. Now the question is how the when nerve impulse is not propagating that time, how the neuron is maintaining this resting membrane potential, how this neuron is maintaining minus 70 MV. So to maintain this resting membrane potential, three factors are involved. Okay, one factor is K plus leak channel and Na plus leak channel. Now in the figure, the K plus channels are shown with the blue color and the Na plus leak channel are shown with the rust color. And in the figure you will observe that in the plasma membrane of neuron, more K plus leak channels are present compared to Na plus leak channels. So this is the figure, and this is the plasma membrane, and here you can see more number of K plus leak channel present compared to sodium Na plus leak channel. Now, what this K plus leak channels are doing? See, as more number of K plus channels are present, so we can understand that more K plus ion leaving the cell, more K plus ion leave the neuron. Okay, and they are expelled out in the extracellular fluid. So as more K plus from the cytosol, more K plus are going outside. So due to that, inside the negative charge develops, and as K plus are coming out, so due to that, on the outer surface of plasma membrane there is a development of the positive charge. Okay. Now, second factor which is maintaining this resting membrane potential are the trap anions. Trap anions are the negative charge ion. And see, this trap ion meaning that we know that the K plus are going out. Okay, but with this K plus, this anion or the negative charge ion, they are remaining inside in the cytosol. They are not going outside because they are attached with the molecule. So negative ion, they are attached with the ATP and also with the large protein. So that's why the negative ion cannot leak out. Okay, so here you can see the phosphate ion is shown, and with the protein, as you can see, the minus sign is shown. That means the negative ions are attached with the proteins. Okay, so this is the second factor which is maintaining the resting membrane potential, and the third factor is Na plus K plus ATPase. Now here this is the Na plus K plus ATPase, and as you can see, this Na plus K plus ATPase is expelling 3 Na plus outside the neuron, and this Na plus K plus ATPase is making the entry of 2 K plus inside the cytosol of the neuron. Okay, so basically what this Na plus and K plus ATPase is doing, so it is balancing the Na plus and K plus outside and inside, and it is also balancing the charge, and for this it will require energy, and this energy is supplied by ATP. ATP is converted into ADP, and ATP provide energy for this active transport of Na plus and K plus. So Na plus K plus ATPase is responsible for expelling 3 Na plus outside, and this Na plus K plus ATPase is bringing 2 K plus inside the cytosol of neuron. Okay. so these are the three factors which are maintaining the resting membrane potential. Okay, so can go through this figure. Okay, now I would like to show you some figures so that you can understand exact mechanism where the nerve impulse generate, how it is generate, and how the nerve impulse propagate. Okay, what is the origin point for the nerve impulse and where it is propagating. So in this figure here, as you can see, the plasma membrane of neuron is shown, and with the purple color they are showing the sodium channel, and this sodium channel are voltage gated Na plus channel. In the first box, as you can see, the channel is closed. Okay, resting membrane potential, outside positive, inside negative. And in the middle box you can see the Na plus channel open, activation. Okay, and in the final box you can see inactivation, closed. Okay, so the Na plus channel, position of Na plus channel. Okay, close, open, closed, resting, activation, inactivation. Now here this is the one neuron is shown, and in the different regions of this neuron you will find different channels. For example, the first segment they are showing the receptive segment, then the second segment is known as the initial segment. This is generally the segment from where the nerve impulse is start to propagate. Okay, axon hillock region. Then see, on the entire length of the neuron, these three channels are present: Na plus, K plus pump, then Na plus leak channel, and K plus leak channel. And this is the conductive segment, this one. And in conductive segment, you can see voltage gated Na plus channel and voltage gated K plus channel are present. Now here the axon terminal are shown, and we know that at the end of axon terminal synaptic end bulbs are present. So in the previous lecture we have discussed about the synaptic end bulb, and we know the synaptic end bulb, they are surrounded by the extracellular fluid, and in that extracellular fluid we know more number of Ca2 plus ions are present. So the concentration of calcium ion is very high in extracellular fluid. So when the nerve impulse reaches to this axon terminal, when the nerve impulse finally reach to the level of synaptic bulb, that time this voltage gated calcium channel open, and calcium enters into the synaptic end bulb. Okay, so this thing we have discussed in our previous lecture. Okay. Now this, see, this figure is showing neuron at rest. So you can go through this figure, see, and see over here outer, on outer surface, as you can see, they are writing the plus sign, and inside minus. Okay, so this is the resting membrane potential, and minus 70 MV is the resting membrane potential. And in the plasma membrane you will find the Cl minus channel, then you will find the potassium channel, then you will find the sodium channel, K plus channel, Na plus channel. Okay, then in the synaptic end bulb you will find voltage gated calcium channel, then the synaptic knob, synaptic end bulb they are showing. Okay, then these are the dendrites. Okay, then receptive segment. So entire neuron is divided into four parts: receptive segment, initial segment, conductive segment. So in this conductive segment, what happens? The nerve impulse is propagating, and final is the transmissive segment, last segment. Now this is the resting membrane potential in the voltmeter. As you can see, resting membrane potential is minus 70 MV, and you see where the electrodes are immersed. Okay, one electrode, the tip of one electrode is present inside the cytosol, and second one is present in the extracellular fluid on the surface of plasma membrane. Okay, and now with the help of voltmeter, the resting membrane potential when measured, it is indicating minus 70 MV value. Okay, now in this figure also a neuron is shown, the different parts of neuron: receptive segment, then the initial segment, conductive segment, and finally the transmissive segment. They are showing overview of events in each neuron. Yeah. Now here, as you can see, one neuron is the postsynaptic neuron, and there are many presynaptic neuron. Okay, and the axon terminal and the synaptic end bulb of this presynaptic neuron, they are forming the synapse with the postsynaptic neuron. So now we are discussing about this. See, postsynaptic postsynaptic potential in the receptive segment. Now, the presynaptic neuron, we know they release the neurotransmitter. In our previous lecture, we learned that the synaptic vesicle, the exocytosis, they burst and the neurotransmitter acetylcholine was released. Okay, so now this where these neurotransmitters are going, so the neurotransmitter, they cross the synaptic cleft, and they join with the receptor. Okay, receptors are present in the plasma membrane of postsynaptic neuron, and when the neurotransmitter bind with the receptor, they initiate a graded potential in postsynaptic neuron. Now binding of this neurotransmitter with the receptor can be either an excitatory or can be inhibitory for post synaptic neuron. So if the neurotransmitter is excitatory, so that time the neurotransmitter will be responsible for an excitatory postsynaptic potential. Okay, thus the neurotransmitter is responsible for the formation or the generation of excitatory postsynaptic potential, that is known as, in short, EPSP. And if neurotransmitter is joining with the receptor, and if it is inhibiting the postsynaptic neuron, that time it is known as inhibitory postsynaptic potential, IPSP. So depending upon the neurotransmitter release and the specific type of acceptor to which it binds, this EPSP or IPSP are determined. So now let's understand this, how these EPSP are generated. So first point, see, synaptic vesicle burst, exocytosis of synaptic vesicle, neurotransmitter are released from synaptic vesicle. Now this neurotransmitter, they are binding to the receptor which are present on the plasma membrane of postsynaptic neuron, and this receptor are present on the surface of chemical gated cation channel. Okay, and now as the neurotransmitter joins with the receptor, so this chemical gated cation channels, they are open now. So here As you can see with the green color, they have shown the neurotransmitter joins with the receptor, and this chemical gated cation channels are open. Now as the channels are open, what happen, from outside Na plus enters in the postsynaptic neuron very quickly. Okay, Na plus flows into neuron very fast, and and faster than K plus flows out. That means K plus is going outside, but it is going slowly. Okay, so as the entry of Na plus is very fast, so inside of the postsynaptic neuron there is a development of more positive charge. Okay, and this development of more positive charge is known as EPSP. And we know resting membrane potential was minus 70 MV, but due to the entry of this sodium, now the negative charge is decreasing, and the negative charge now becomes minus 68 MV, and this membrane potential minus 68 MV is known as EPSP. Now this EPSP, as you can see, the EPSP propagate toward the initial segment of neuron. So this is about the EPSP. Okay. Now let's understand this generation of IPSP. Now, in case of IPSP, neurotransmitter release from presynaptic neuron. So here with the red color, as you can see, the synaptic vesicle bursting, neurotransmitter are releasing, neurotransmitter bind to the receptor. Okay, so now the chemical gated K plus channel or chemical gated Cl minus channels are open, and as these channels are open, so now what happened, either K plus is moving out of the postsynaptic neuron, it is flowing outside, or it is possible that the Cl minus negative charge ion are entering inside the postsynaptic neuron. Okay, so inside of the postsynaptic neuron becomes more negative because K plus went outside, they are expelled outside, and there is the entry of Cl minus inside the postsynaptic neuron. So that's why the membrane potential becomes more negative and it is having value of minus 72 MV, and this value is known as IPSP. So now in postsynaptic neuron, IPSP is going to propagate in the initial segment of the neuron. So here we have discussed about this thing. Okay, okay, now we are discussing about, so we have with this we have completed our discussion on resting membrane potential. Okay, now we know that resting membrane potential is minus 70 MV, and now we are going to discuss about the action membrane potential. So according to the theory, the process of nerve impulse conduction, we can divide the propagation of nerve impulse in two main phases. One phase, resting membrane potential, we know that, and second phase is the action membrane potential. Now, now transmission of nerve impulse along the nerve fiber, also known as conduction of or the propagation of nerve impulse throughout the axon, along the axon. So transmission of signal by a nerve occur in four phases. One is initiation of the impulse. Second is transmission along a nerve fiber. Third is transfer of nerve impulse into target muscle or nerve. And finally, the nerve impulse will reach to the target tissue. Okay, and the target tissue cell, they are going to start functioning now. So the first phase is the depolarization phase. Okay, so now we are discussing this depolarization process. Now here in this figure, this is the point. Okay, they are showing. Now in this figure, see action potential, events associated with the depolarization. So in the first figure, see this small graph you can see, and in this graph, see on y-axis they are showing the membrane potential mv, and on x-axis they are showing the time, okay, in millisecond. So during resting, we know the resting membrane potential is minus 70 mv. Okay, and during this period, as you can see, on the plasma membrane of neuron, outside there is a positive charge and inside there is a negative charge. And voltmeter is showing the value minus 70 mv. Minus 70 mv is a resting membrane potential, and at this time, at this point, see Na plus channel, so voltage gated Na plus channel closed during resting membrane potential. Now what happens, what happen when the neuron is depolarized? Okay, so now in this figure, as you can see, now the voltage gated Na plus channel is open, it is activated. So now Na plus flows inside. Okay, Na plus, we know Na plus are positively charged, Na plus flowing inside from adjacent region, and the threshold value is reached. So minus 70 is the resting membrane potential, and now as the Na plus are coming inside the cytosol, what happened, this negative value is decreasing and it is reaching to the level of minus 55, and this minus 55 is known as the threshold value for membrane potential. Okay, so in this, voltmeter is showing minus 55 mv. Now see the Na plus channel voltage gated still open, so more and more Na plus are coming in the cytosol. So as more and more Na plus are coming very fast inside the cytosol, so the membrane potential now becomes positive and it attains the value of plus 30. Okay, so inside the plasma membrane there is a development, as you can see, there is a development of positive charge due to sodium entry, and here on outside there is a development of negative charge. So now we can say the neuron is depolarized. Okay, and after depolarization, once the depolarization state is achieved, so now the Na plus channel, they are closed. So the further entry of Na plus inside the cytoplasm is prevented, and now the membrane potential is having a value of plus 30 mv. This is the peak value. Okay, so here you can see outside negative, outside negative, inside positive. So this is known as the depolarization. Okay, resting membrane potential. Okay. Now in this, see first, uh, I would like to show you this figure. Okay. Now this is the resting state of the neuron, and in the resting state you can see the resting membrane potential value minus 70 MV. Okay, and now during resting period, resting state, what happened? So voltage gated K plus and Na plus channel are closed. So this channel is closed, this channel is also closed. Okay, so the axon plasma membrane is at resting membrane potential. Small buildup of negative charge along inside surface of membrane. So here you can see there is the presence of negative charge. Okay, and equal buildup of positive charge outside the surface of membrane. So during resting period you can see here there is a positive charge present, and inside there is a presence of negative charge during the resting state, and during resting state Na plus and K plus channel are closed and membrane potential is minus 70 MV. Now what are the changes, what are the event that are taking place during depolarization? So now during depolarization, see first of all, you observe this channel. See in the green color sodium channel, there are two gate. One is activation gate, this one, and second gate, this gate is known as the inactivation gate. So there are two gate in Na plus channel. So now during depolarization, what is happening? See, when the membrane potential of axon reaches the threshold, that is the minus 55 MV, that time the Na plus channel activation gate open. So as the activation gate, as you can see here, the activation gate is open. So now from extracellular fluid, Na plus is entering into the cytosol very quickly. So inside in the cytoplasm there is a buildup of Na plus, that is the buildup of positive charge. So here you can see the development of positive charge, and outside now there is a negative charge. Okay, so we can say now the membrane is becoming depolarized. Okay, so now see, so due to the depolarization, the membrane potential uh, is reached to the level of plus 30 MV. So this is the peak value, okay, for depolarization, and this value, this plus 30 value, plus 30 MV, for that the entry of Na plus inside the cytosol, it is responsible. So this is about the depolarization. Okay. Now wait a minute, let me scroll. Okay. Now first, let's try to understand this graph. See, on x-axis they are showing time in millisecond. On y-axis they are showing the membrane potential in millivolt. Okay, now this is the minus 70 MV. That means this is the resting membrane potential. Now when the during the depolarization, what happened? Na plus channel open, so Na plus enters in the cytoplasm very quickly. So now the membrane potential become the positive, and the peak value is plus 30. So this plus 30 value is known as the spike, or this is the depolarization peak value, or this value is also known as the action potential. So now from this value, from this point, the nerve impulse start propagating. So depolarization wave is going to spread in the entire length of the neuron. So now see, once the event of depolarization has occurred, once this plus 30 MV value is achieved, so the nerve impulse or the spike is initiated. This is the initiation of nerve impulse, this point. Now what is action potential? What is nerve impulse? See, action potential is the another name of nerve impulse, and this nerve impulse propagate throughout the membrane of the neuron from one point to the farthest point. Okay, so we can say simply, the nerve impulse propagate in the entire length of the plasma membrane of neuron, and finally the nerve impulse reaches to the far end of the neuron. Now the action potential lasts for about 1 millisecond. The duration of action potential is 1 millisecond, and the negatively charged point, the depolarized region, what happened, we know in depolarization there is a development of negative charge outside. So when there is a negative charge present outside, that region affect the neighboring region. So now the neighboring or the adjacent portion also become depolarized. So now this depolarized adjacent portion is going to depolarize the neighboring adjacent portion. The neighboring adjacent portion is going to depolarize. Now this depolarized region is again going to depolarize the adjacent region. So this way the entire depolarization wave is propagating in the entire length of the neuron. So as now the value of action potential, see the action potential value. So now the resting membrane potential was minus 70, and now the membrane potential become plus 30 when the action potential uh, is generated and action potential is propagating now. Okay, now depolarization and reversal of potential requires only zero point 0.5 millisecond time. Okay. So now here, see here, propagation of action potential, all the information, you can go through this information. Okay. Events of propagation of action potential, okay, 1 2 3, and this is the fourth point. Okay, and now let's try to understand how this depolarization is spreading. Okay, how the nerve impulse propagate, how the nerve impulse is conducted. So here you can see this portion, this part, this part is depolarized. Now as this portion is this depolarized, that means the sodium channel were open, Na plus enters inside the cytosol very quickly. Cytosol also known as exoplasm, plasma membrane, exolemma. So now there is a development of negative charge outside, inside positive, because Na plus is there in exoplasm. Now this negative charge portion, this region affects the neighboring portion. So now this part become depolarized. So here in this region, what happened? The Na plus channel open. So now Na plus enters here. So now this portion depolarized. So now this portion is going to affect this portion. Okay, so here now there is going to Na plus channel open. So Na plus entering. So now that portion is depolarized. Okay, so now this depolarized region is going to affect this region. Okay, so now in this region Na plus channel are open, Na plus enter inside. So now this portion is going to become depolarized. Okay, so finally this portion outside negative, inside positive, due to the entry of Na plus inside. So this is how the action potential propagate. Okay, 1 2 and 3 and 4, propagation of action potential. Now we are discussing about the repolarization. Okay, so the depolarized region now going to become repolarized. So what are the events that are taking place during repolarization? So see, this is the peak point, action potential generated, depolarization take place at the value of plus 30 due to the influx of Na plus inside. Okay, this is the plus 30. Now see, first repolarization. Now in the repolarization process, what happened? In the repolarization, see, this is the K plus channel, voltage gated K plus channel. Okay, and this channel are open now. So now K plus from cytosol is moving outside, and K plus enters in the interstitial fluid. Okay, so as K plus are moving outside, so during the depolarization the value was plus 30, but now as the K plus are going outside, so the positive charge is moving outside. That's why again the membrane potential becomes negative, and it attains the value of minus 70 once again. Okay, so once again during repolarization K plus are going out, and due to that this plus 30 MV value, once again it becomes negative minus 70 MV. Now it is not that when the minus 70 value is attained, that time the K plus are going to stop. Even though minus 70 MV potential is achieved and repolarization occur, outside positive, inside negative, still what happen, more and more K plus are still continue to go outside. They are moving outside, and as more K plus are going outside, so the membrane potential becomes more negative, so it attains the value of minus 80 to minus 90 MV. Okay, so this value minus 80 to minus 90 MV, that value is known as hyperpolarized. Okay, now after hyperpolarization, the resting membrane potential is re-established. So now which are the event, how the so once during repolarization, once the value reached to the level of minus 90, and how once again this minus 70 membrane potential is maintained, that is the question. So how the RMP, resting membrane potential, is re-established? Which are the events? Okay. So once again, let's try to understand this with another figure. Repolarization, another figure we are looking at. So see, okay, repolarization phase begin. Now in repolarization, what is happening? See, in depolarization, value reach plus 30. Okay, now action potential generated, it is now started to propagate. Okay, and now the repolarization begins. Now what happened in repolarization? So Na plus channel inactivation get closed. Here you can see inactivation get is closed and K plus channel open. So here you can see the K plus channel is open. Now the membrane start to become repolarized. Why the membrane is becoming repolarized? Because the K plus ion leaving the neuron. So from the cytosol they are entering into the interstitial fluid or the extracellular fluid. Okay, so K plus, in short, they are moving outside. So now there is a drop, and once again this membrane potential is start to become, it start to falling. Okay, and it is moving toward the resting membrane potential. Now this repolarization is continuing. So as the repolarization continuing, more and more K plus flowing outside continuously, more K plus ion leaving the neuron, more negative charge build up along the inside surface of the plasma membrane. Okay, now K plus outflow eventually restore the resting membrane potential, that is minus 70 MV. Okay, and sometimes as more K plus are going outside, due to that there is a development of more negative charge. Okay, and after that repolarization, so finally the neuron attains resting state. So I am showing you this entire figure. Okay, now one more. See, when the when the K plus moving outside, that is the repolarization process, voltage gated K plus channel are open and K plus are flowing outside. But as I told you that the K plus, the movement of K plus is very fast, and these K plus channel are still open, so more and more K plus are going outside. So even though minus 70 MV resting membrane potential was achieved, but as the more K plus are going outside, so due to that the membrane potential becomes more negative and it attains the value of minus 90 MV. So this minus 90 MV value is known as after hyperpolarization phase. Okay, now in this phase what is happening, the K plus channel, now they are going to close once this much membrane potential is achieved. Now the K plus channel are going to close, and as the K plus channel are closed, membrane potential return to the resting level of minus 70 MV afterwards. Okay, now unlike the voltage gated Na plus channel, most voltage gated K plus channel do not exhibit inactivated state. Instead they alternate between the closed and open state. Now what is this? See, so the question is, even if the membrane potential has reached to the level of minus 90 MV, so how the resting membrane potential minus 70 MV is restored? That is the question. Okay, so now let's try to find out that answer. See, now the repolarization period return the cell to its resting potential. So in the repolarization, the value changes from plus 30 to minus 70. Okay. Now the neuron is now ready to receive once the minus 70 value resting membrane potential once again the repolarization. So now the neuron is again ready to receive another stimulus, and neuron is now ready to conduct or propagate another nerve impulse. Now, in fact, until repolarization occur, as long as the repolarization is taking place, neuron cannot conduct another impulse. So that's why it is now becoming necessary to restore the normal resting membrane potential. And how this normal resting membrane potential is going to maintain? So by expelling the Na plus ion outside and taking back the K plus ion inside. So who is going to restore this? So sodium potassium pump, they are start working, and they are throwing back the Na plus outside, and they are bringing K plus inside the cytosol. So now the resting membrane potential is achieved once again. So during this exit of Na plus and entry of K plus, the time taken for this event is known as the refractory period. And because during this period the membrane is incapable of regenerating another impulse. So during this period the nerve impulse cannot propagate, it cannot generate. Now, one more benefit of refractory period is that the impulse travel in the axon in only one direction. Okay, so in entire length, the nerve impulse is propagating in only one direction. So the nerve impulse is unidirectional. Okay. Now the refractory period is very short, and being only about 1 millisecond. A refractory period is 1 millisecond, and thus a nerve fiber can transmit about 1000 impulses per second. Okay. So we have discussed all the theory point. Now I would like to show you all the figures. Okay. Now this we have seen. Now this is here they are showing the different channels, okay, different types of channels. So with this we have completed this lecture on generation conduction of nerve impulse. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.